Genomic imprinting is a fascinating biological process. It means that some genes in our bodies are expressed differently. This difference depends on which parent they came from. For most genes, we inherit two working copies. One copy comes from our mother and one from our father. However, with imprinted genes, only one of these two copies is active. The other copy is silenced or turned off. This parent of origin specific gene expression is vital. It plays a critical role in our development. Understanding this process is key to understanding certain genetic disorders. It's a fundamental aspect of mammalian genetics. The requirement for genomic imprinting underscores the importance of contributions from both parents. Normal development in mammals relies on having both a maternal and a paternal set of chromosomes. These chromosomes carry specific imprints. These imprints are established in the eggs and sperm. They ensure that certain genes are expressed from only the mother's chromosome, while others are expressed only from the father's. This delicate balance is essential for growth. It is particularly important for fetal growth and placental development. If this balance is disturbed, serious developmental issues can arise, highlighting the precision required by nature. When the process of genomic imprinting goes wrong, it can lead to specific disorders. These are known as imprinting syndromes. These conditions occur when the expression of an imprinted gene is abnormal. For example, a gene that should be active might be silent, or a gene that should be silent might be active. Sometimes, both copies of a gene might be active, or both might be silent. These disruptions highlight the critical roles that imprinted genes play. They show how a precise dosage of gene products from these specific loci is necessary for health. These syndromes are often complex, affecting many systems in the body. Genomic imprinting is a feature primarily found in placental mammals. It is also observed in some marsupials. However, it is notably absent in other vertebrates like birds or egg-laying mammals such as monotremes. This evolutionary pattern suggests that imprinting co-evolved with placental development. Imprinted genes are known to influence a variety of traits. These include birth weight, as seen with genes like AND. They also play roles in metabolic functions and even behaviors. For instance, some imprinted genes affect suckling and feeding behaviors in newborns, ensuring their early survival and growth. The study of these genes continues to reveal their widespread impact. Epigenetics provides the molecular basis for genomic imprinting. Epigenetic marks do not change the DNA sequence itself. Instead, they are chemical modifications to DNA or its associated proteins. These marks act like switches or tags. They tell the cellular machinery whether an imprinted gene should be turned on or off. This marking process occurs differently in sperm and eggs. This ensures that the maternal and paternal alleles have distinct epigenetic signatures. These signatures are then maintained in the developing embryo. This system allows for the parent-specific gene expression that defines imprinting. It's a sophisticated layer of genetic control. DNA methylation is a primary epigenetic mechanism in imprinting. This process involves adding a small chemical group, called a methyl group, to DNA. Specifically, it is added to cytosine bases, often in regions called CpG islands. When these regions are heavily methylated, the gene nearby is usually silenced. For imprinted genes, one parental allele will be methylated while the other is not. This differential methylation pattern is established in the germ cells. It is then faithfully copied during cell division throughout development. This ensures the imprinted status is maintained in somatic cells. It is a stable and heritable mark crucial for imprinting. Histone modifications are another vital epigenetic mechanism. Histones are proteins that package DNA into a compact structure called chromatin. Various chemical modifications can occur on histone tails. These include acetylation, methylation, and phosphorylation. These modifications can alter how tightly DNA is wound around the histones. Looser chromatin generally allows genes to be active. Tighter chromatin usually silences genes. Specific patterns of histone modifications are associated with active or silent imprinted alleles. These work in concert with DNA methylation to regulate gene expression from the correct parental chromosome. They add another layer of complexity to the epigenetic code. Non-coding RNAs also play a significant role in controlling imprinted genes. Long non-coding RNAs, LNCRNAs, are particularly important. These RNA molecules do not code for proteins. Instead, they can act as regulatory molecules. Some LNCRNAs are transcribed from imprinted regions. They can then silence other genes in the same region. often on the same chromosome they are transcribed from. A well-known example is the lncRNA involved in Angelman syndrome. It silences the paternal copy of. Researchers have also identified non-canonical imprinting mechanisms. These are independent of DNA methylation, suggesting even more diverse control pathways. Angelman syndrome, AS, is a severe neurodevelopmental disorder. It provides a clear example of what happens when imprinting goes awry, as is primarily caused by a loss of function of the maternally inherited gene. This gene is located on chromosome 15. The gene codes for an enzyme involved in protein degradation. This enzyme is particularly important in the brain. Understanding the genetic basis of AS has been crucial. It has paved the way for research into potential therapies. The syndrome typically becomes apparent in early childhood. It presents a range of characteristic features. The genetic mechanism underlying Angelman syndrome is quite specific. In certain brain regions, only the maternal copy of the gene is active. The paternal copy of is normally silenced. This silencing is achieved by an antisense long non-coding RNA called. This lncRNA is transcribed from the paternal chromosome. It effectively shuts down the paternal gene. Therefore, if the maternal copy of is lost or mutated, neurons in these brain areas have no functional UBE3A protein. This deficiency leads to the symptoms associated with Angelman syndrome. It highlights the vulnerability caused by monoallelic gene expression. Individuals with Angelman syndrome exhibit a distinct set of clinical features. These often include severe developmental delays and intellectual disability. Speech impairment is common, with many individuals being nonverbal or using very few words. Motor difficulties, such as problems with balance and coordination, ataxia, are also characteristic. Many individuals with AS experience seizures. Despite these challenges, they often have a happy demeanor. They may display frequent laughter and excitability. These features, along with specific physical characteristics, help clinicians diagnose the syndrome. Early diagnosis is important for appropriate management. The Imprinting Center, IC, plays a crucial role in this process. This genetic region, located near, controls the imprinting status of several genes on chromosome 15. It regulates the expression of. Therefore, the IC dictates whether the paternal allele will be silenced. Defects in this imprinting center can also lead to Angelman syndrome. This occurs if the center fails to establish the correct maternal imprint. In such cases, the maternal might behave like a paternal allele and be silenced. This further illustrates the complexity of imprinting regulation. Research continues to explore these intricate control mechanisms. Recent advancements in epigenetic editing offer new hope for Angelman syndrome. Scientists are developing tools that can modify epigenetic marks at specific gene locations. One such promising tool is known as dCas9 SDD. This system is based on the CRISPR-Cas9 technology. However, it uses a dead Cas9, dCas9 protein. This dCas9 cannot cut DNA. Instead, it is fused with other proteins that can alter epigenetic marks. For Angelman syndrome, the goal is to reactivate the silenced paternal gene. This approach targets the root cause of the disorder. The dCas9 SDD system works in a targeted manner. It is designed to recruit DNA methyltransferases. These enzymes add methyl groups to DNA. The system guides these enzymes to the IC locus on the paternal chromosome. Methylating this imprinting center is expected to silence the long non-coding RNA. If is silenced, it can no longer suppress the paternal gene. This would effectively unsilence or reactivate the paternal allele in neurons. This would restore UBE3A protein levels in the brain. This strategy is a sophisticated form of gene therapy. A study published in September 2024 demonstrated the potential of this approach. Researchers used the dCas9 SDD tool in a mouse model of Angelman syndrome. They observed increased DNA methylation at the IC. This led to reduced expression of and, crucially, paternal expression was restored to significant levels in various brain regions. These molecular changes translated into behavioral improvements. The treated AS mice showed better motor skills, balance, and exploratory activity. These results are highly encouraging for future clinical applications. The efficiency of methylation was also notably high. An important aspect of this new technology is its specificity and safety. The dCas9 SDD system showed minimal off-target effects in the mouse study. This means it primarily modified the intended genetic locus. Whole genome analysis confirmed no significant off-target methylation. This is a major advantage over some other gene therapy approaches. It avoids making permanent breaks or changes in the DNA sequence itself. Instead, it alters gene expression by changing epigenetic marks. This targeted DNA methylation offers a potentially safer and reversible therapeutic strategy for Angelman syndrome. Further research is needed to translate this to humans. Section 5. Prader-Willi syndrome. Understanding the paternal absence. Prader-Willi syndrome, or PWS, is another well-known imprinting disorder. Like Angelman syndrome, it involves genes on chromosome 15. However, PWS results from the loss of function of paternally expressed genes in the 15q11q13 region. In this case, the maternal copies of these genes are normally silenced by imprinting. Therefore, if the paternal copies are missing or non-functional, the individual lacks the necessary gene products. PWS is a complex disorder. It affects multiple systems in the body. It presents with a range of physical, cognitive, and behavioral symptoms. Understanding its mechanisms is vital for management. The clinical features of Prader-Willi syndrome change throughout an individual's life. In infancy, babies with PWS often exhibit severe hypotonia or low muscle tone. They may have feeding difficulties and fail to thrive. However, this phase is typically followed by a later stage, usually starting in early childhood. This later stage is characterized by hyperphagia. This is an insatiable appetite that leads to chronic overeating and a high risk of severe obesity. Individuals with PWS also commonly experience intellectual disabilities. They may have learning difficulties and behavioral problems, such as temper outbursts and obsessive-compulsive traits. The genetic basis of PWS is centered on the absence of paternal gene expression from the 15q11q13 region. This can occur through several mechanisms. The most common is a deletion of this region on the paternal chromosome. Another cause is maternal uniparental disomy. This means the individual inherits 2 copies of chromosome 15 from the mother and none from the father. Less commonly, PWS can result from a defect in the imprinting center. This defect would prevent the correct paternal pattern of gene expression from being established. Regardless of the specific genetic cause, the outcome is the same. There is no active contribution from the critical PWS genes. Managing Prader-Willi syndrome presents significant challenges. There is currently no cure. Treatment focuses on managing symptoms and improving quality of life. Growth hormone therapy can help improve growth, body composition, and muscle tone. Strict dietary management and supervision are essential to control hyperphagia and prevent obesity. Behavioral therapies and educational support are also important components of care. The constant hunger and associated metabolic complications remain major hurdles. This underscores the need for continued research into the underlying biology of PWS. New insights could lead to more effective interventions for this challenging disorder. Section 6. The rhythmic genome DNA replication timing in Prader-Willi syndrome. Recent research has shed new light on the PWS locus. It involves a fundamental cellular process, DNA replication timing. During the S phase of the cell cycle, DNA is duplicated. It turns out that not all parts of the genome replicate at the same time. Active genes and gene regions tend to replicate earlier in S phase. Silenced regions often replicate later. A study published around October 2023 investigated this in the context of imprinted genes. The researchers used human embryonic stem cells with either 2 maternal or 2 paternal genomes. This allowed them to compare replication timing. The findings from this study were quite striking. The PWS locus exhibited the most extreme replication asynchrony based on parent of origin. This means there was a very large difference in when the paternal and maternal copies of this region replicated. Specifically, the active paternal allele at the PWS locus replicated much earlier than the silenced maternal allele. This difference spanned a large region of nearly 3 million base pairs. The maximum replication timing difference was substantial. This asynchrony was not limited to stem cells. It was also maintained when the cells were differentiated into neuronal precursor cells. This correlated with the known gene expression patterns. The replication asynchrony observed was significant. The study found that the PWS locus showed a mean difference of 1.67 standard deviations across the region. This indicates a consistent and robust parent of origin effect on replication timing. Researchers also looked at other known imprinted gene clusters. These included the DLK1DEO3, H19/IGF2, and GNAS loci. These regions also showed differential replication timing. The active alleles in these clusters replicated earlier. However, the effect at the PWS locus was particularly pronounced. This suggests a strong link between imprinting status and the timing of DNA duplication for these genes. These discoveries have important implications for understanding genomic imprinting and PWS. The study suggests that replication timing may be an integral part of the biology of imprinting. It might help establish or maintain the epigenetic marks that define imprinted genes. It could also influence chromatin structure and gene accessibility. While the vast majority of the genome, 99.82%, showed no significant replication asynchrony, these specific imprinted loci stood out. Further research is needed to understand how replication timing contributes to the PWS phenotype. It may also offer new perspectives for potential therapeutic interventions in the future. It's a new layer in the complex regulation of imprinted genes. Section 7. Beyond Humans, Imprinting Disorders in Livestock. Genomic imprinting is not unique to humans. It is a conserved phenomenon in placental mammals. This includes many livestock species. Animals such as cattle, sheep, and pigs also have imprinted genes. Disruptions in these genes can lead to imprinting-like disorders in these animals. Studying these conditions in livestock provides valuable insights. It can help us understand the fundamental mechanisms of imprinting. It also highlights the economic impact of these disorders in agriculture. Livestock models can be very useful for research. They often share physiological similarities with humans. Over 30 imprinted genes have been validated in these species. One example of an imprinting disorder in livestock is large offspring syndrome, or LOS, in cattle. LOS is often observed in calves produced by assisted reproductive technologies, like in vitro fertilization. These calves are typically oversized at birth. They may have other developmental abnormalities. LOS shares similarities with the human Beckwith-Wiedemann syndrome, or BWS. Both conditions involve dysregulation of imprinted genes in critical growth-related domains. These include the IGF-2H19 and CDKN1-CKCNQ1 regions. Loss of imprinting in these areas affects fetal growth and development. This shows how conserved these imprinted gene functions are across species. The callipyge phenotype in sheep is another fascinating example. Callipyge means beautiful buttocks in Greek. It refers to a condition causing extreme muscle hypertrophy, particularly in the hindquarters. This trait is inherited in a unique way called polar overdominance. It only manifests if the mutation is inherited from the father. The mutation is located in the DLK1-MEG3 imprinted region. It leads to overexpression of certain imprinted genes, including DLK1 and PEG11 in muscle tissue. In pigs, a different mutation in the IGF2 gene, inherited paternally, also causes hypermuscularity. This mutation doesn't affect DNA methylation. Instead, it alters the binding of a transcriptional repressor. Livestock species serve as important animal models for human imprinting disorders. Pigs, in particular, are considered promising models. This is due to their physiological and anatomical similarities to humans. Their genome is also well characterized. Modern genetic engineering tools such as CRISPR/Cas9 can be used in pigs. This allows researchers to create specific models of human imprinting syndromes. By studying these animal models, scientists can learn more about disease mechanisms. They can also test potential therapeutic strategies. This comparative approach broadens our understanding of imprinting across the mammalian class. It aids in developing solutions for both human and animal health. Section 8. Translating Discoveries, Real-World Implications and Challenges. The growing understanding of imprinting syndromes has significant real-world implications. Advances in genetic testing have improved diagnostic capabilities. Families affected by these complex disorders can receive more accurate diagnoses. This often happens earlier than in the past. This knowledge is crucial for genetic counseling. It helps families understand the inheritance patterns and recurrence risks. Furthermore, a better grasp of the underlying molecular defects can guide management strategies. It allows for more targeted supportive care. This can improve the quality of life for individuals with these conditions. Patient advocacy groups also play a vital role in this translation process. However, developing effective treatments for imprinting syndromes faces numerous challenges. The epigenetic regulation involved is incredibly complex. These disorders often affect the brain, a difficult organ to target therapeutically. achieving precise targeting of epigenetic therapies to the correct cells and genes is a major hurdle. There's always a risk of off-target effects with gene editing or epigenome editing tools. These could have unintended consequences. The timing of intervention is also critical. Many imprinting disorders have neurodevelopmental origins. This means that treatment may need to be administered during specific developmental windows to be effective. These are substantial obstacles to overcome. The prospect of epigenetic therapies also raises important ethical considerations. Distinctions must be made between somatic cell editing and germline editing. Somatic therapies affect only the individual being treated. Germline modifications would be heritable, passing changes to future generations. The latter raises more profound ethical concerns. Long-term safety of any novel therapy is paramount. We need to ensure that treatments do not have unforeseen adverse effects years later. Equitable access to these advanced and potentially expensive therapies is another societal challenge. We must consider how to make these treatments available to all who need them. These discussions are ongoing. Continued research is absolutely essential to move the field forward. There is still much to learn about the fundamental mechanisms of genomic imprinting. We need a deeper understanding of how epigenetic marks are established, maintained, and erased. More research is needed on the specific roles of different imprinted genes. We also need to study the long-term consequences of manipulating epigenetic states. This includes understanding the stability of any therapeutic changes. Basic science discoveries will continue to fuel the development of new diagnostic tools. They will also inspire novel therapeutic approaches for these challenging disorders. Collaboration between researchers and clinicians is key. Section 9. The future of healing epigenetic therapies for imprinting syndromes. The future of treating imprinting syndromes looks increasingly hopeful. This optimism is largely due to breakthroughs in epigenetic therapies. The development of tools like the DCAS9 SDD system for Angelman syndrome is a prime example. These technologies offer the potential to correct the underlying molecular defects. Instead of just managing symptoms, they aim to restore normal gene function. For Angelman syndrome, reactivating the silent paternal UBE3A gene could be transformative. This precise targeting of epigenetic marks represents a new paradigm in genetic medicine. It moves beyond the DNA sequence to influence gene activity. Beyond sophisticated epigenetic editing, other therapeutic strategies are also being explored. Small molecule drugs could be developed to modulate the activity of epigenetic enzymes. These enzymes are responsible for adding or removing marks like DNA methylation or histone modifications. Such drugs might offer a less invasive way to alter gene expression. RNA-based therapies are another avenue. These could target the non-coding RNAs involved in imprinting. For example, an antisense oligonucleotide could block UBE3A-ATS in Angelman syndrome. Researchers are also investigating strategies to reactivate entire silenced chromosomal regions. The field is dynamic and rapidly evolving. We can envision a future where imprinting syndromes are far more manageable. Continued innovation in gene editing, epigenome editing, and drug discovery holds immense promise. These advancements could lead to treatments that significantly improve the neurodevelopmental outcomes. They could alleviate the debilitating symptoms associated with these disorders. The goal is to provide therapies that not only treat but potentially prevent the most severe consequences of imprinting defects. This would dramatically enhance the quality of life for affected individuals and their families. The pace of discovery suggests this future may not be too distant. Achieving this future requires a concerted and collaborative effort. Basic scientists must continue to unravel the complexities of imprinting. Clinicians are needed to translate these findings into safe and effective treatments. Patient advocacy groups play a crucial role. They support research, raise awareness, and represent the needs of affected families. Regulatory agencies and policymakers also have a part to play. They must ensure that new therapies are rigorously evaluated and made accessible. By working together, the scientific and medical communities can harness the power of epigenetics. This will unlock new ways to heal these profound developmental disorders. The journey is challenging, but the